Okay, so I think my mic is okay, my internet is okay. Okay, so I hope that later connection is okay. No loose connection. Capability. Okay. Okay, so now it's already 8 o'clock. Okay, so we continue start our class now. So for today, we will proceed chapter 5, semester 1, cellular respiration. So last week, I already uh we already teach the glycolysis and also energy, right? Energy carrier. So for today we will continue to Krebs cycle. Okay, so actually if you guys still remember about the reaction, sorry. Okay, so if you guys still remember about the reaction, so actually our aerobic respiration contains three main stages, right? Glycolysis, Krebs cycle, and also electron transport chain. So for today, we will uh, explain about the Krebs cycle and also electron transport chain. So these two are very long topic lah. And also the totally new topic to you. You never learned it before in your SPM. So let's look about it. Okay, so actually what is the Krebs cycle? So actually it has many names. Another name is called citric acid cycle. So if you see the book from Campbell or international book, it will say that it is citric acid cycle. Because why it say it is citric acid? Normally this the Krebs cycle, the origin, the main component is acidic component. So otherwise, but in our syllabus, MPN syllabus is mentioned Krebs cycle. But if you see the books, whatever book, it say citric acid cycle or other tricarboxylic acid cycle, all that, they are also same. So you don't need to be confused. They are totally same cycle, just different name only. So normally Krebs cycle is the process that occur in the mitochondrial matrix. You need to remember glycolysis is at the cytoplasm, right? Krebs cycle is at the mitochondrial matrix. So it will to break down. It need to further oxidize the glucose molecule. So as in our glycolysis, the glucose molecule is actually oxidized, become pyruvate already, right? One glucose molecule can oxidize two pyruvate molecule. And then this pyruvate molecule later will enter Krebs cycle for further oxidation again. So what are the process? So later you will learn about it. So, but before pyruvate enter Krebs cycle, it will be further oxidized first to form acetyl CoA first. This one it must be form acetyl CoA first. Pyruvate cannot directly enter Krebs cycle, so it have the one reaction we call as link reaction. This is the link reaction. So this link reaction will oxidize pyruvate to form acetyl CoA. So this reaction name we call as oxidative decarboxylation. So what is this mean? Oxidative decarboxylation. So as the first term, oxidate. So this means it is oxidation reaction. Pyruvate is oxidized to form acetyl CoA. And then this decarboxylation is meaning that it will remove one carbon dioxide. So if you alert here, this one, if you alert here, this carboxyl group COO, actually this one is carboxyl group, COO. Actually, it will be already be removed, and then the coenzyme A is added. So this reaction actually is very complex reaction. I can say it is very actually it is very complex reaction. So actually you see that this is the general overall equation. So pyruvate will be oxidized to form acetyl-CoA. So the reaction name is oxidative decarboxylation. If you see the middle part, you can see that there are many components, right? So the enzyme that used in this reaction is called pyruvate dehydrogenase complex. So what it means that it is complex? Because actually it contains many enzymes. This reaction is very complex. Actually, there are many intermediate within this reaction. So you see that there are one coenzyme, coenzyme A. This one is one type of coenzyme. We call as coenzyme A. Coenzyme A, it will be substituted into the pyruvate, and then the carbon dioxide will be removed. The carbon dioxide from the pyruvate will be removed, and then at the same time, your our energy carrier NAD plus will be reduced to form NADH and hydrogen ion, and then they are also need to use DPP, FAD, magnesium ion, and lipoic acid. So these are the overall reaction only. But in detail, so you see here this diagram. So this diagram is detailed. How pyruvate can be oxidized to form acetyl-CoA. Acetyl-CoA. So acetyl-CoA is this. This is our final product. And also, what are the intermediate? So there are four main reactions here. And then there are three main enzymes actually will be covered. So the general name is just pyruvate dehydrogenase complex. So what is called complex actually is very huge complex. The enzyme is huge complex. So there are total three types of enzyme will be used. So pyruvate dehydrogenase, dihydrolipoic acetylase, dihydrolipoic dehydrogenase. So there are many lah. So the first reaction is pyruvate will lose carbon dioxide and HETTP is formed. Okay, so you will see here HETTP is this lah, is this structure, hydroxyl ethyl TPP. So TPP actually is thiamine biophosphate. So the pyruvate will remove carbon dioxide gas, and then the remain carbon chains, these two carbon will be substituted by thiamine biophosphate. So thiamine biophosphate will be substituted into the origin pyruvate molecule lah. So this one reaction is used, enzyme is used pyruvate dehydrogenase. So it will form hydroxyl ethyl TPP. This is the IUPAC name lah, hydroxyl ethyl, because it contains hydroxyl group and also ethyl group. So it use this lah. But actually this one is not I can say it's common name lah, not IUPAC name lah. Common name. So HETTP. Okay. After that, this hydroxyl ethyl TPP will be transferred to lipoic acid and oxidized to form acetyl dihydrolipoamide. It will be oxidized. It will go to the oxidized to form the acetyl dihydroxylipoamide. So it will combine with this lipoic acid. So this one is the structure of lipoic acid lah. This is the structure of lipoic acid. Okay, once it combines with the this structure combines to form this structure, later acetyl group is transferred to CoA. So this one, the acetyl group. So if you see here, this is the we call as alcohol, right? So when alcohol undergo oxidation process, it will form carbonyl group, right? But this one is for semester 3 lah. Semester 3 senior, actually you should know the oxidation of alcohol will form a carbonyl group, right? But for sem 1 you don't need to know. So this one is called as oxidation. So this oxidation process, later lipoic acid will bind to this acetyl group. This one actually we own as acetyl structure already. Acetyl structure. So acetyl group, so this acetyl group will be transferred to the coenzyme A. So it will bind with the coenzyme A to form acetyl-CoA. So why it is known as acetyl-CoA? Because it contains one acetyl group and also coenzyme A here. And then and then and then and then dihydroxyl lipoamide will be reoxidized again to form reoxidized again to form the NADH to provide the energy to NAD plus, and then so that the NAD plus will be reduced to form NADH. So there are two enzymes here. So the fourth reaction is the dihydroxyl dehydrogenase, and another one middle action is middle one. This step 2 and step 3, the enzyme used is dihydroxyl lipoic transacetylase. Okay, so this is the overall of the link reaction. This not overall lah, detail of the link reaction. So this one actually you will take you can learn in detail in biochemistry books. Biochemistry book will explain in detail. But in Campbell, I think no, don't. Campbell just say like this only, right? So okay, then so you need to know it because it is the detail in the syllabus. You need to know the link reaction. Okay, yeah, but the most important is you need to know the enzyme name lah. Pyruvate you need to use at least you need to know this lah. Pyruvate dehydrogenase complex and what are the substitute group, coenzyme A, and carbon dioxide release, and also got one NADH produced. So in link reaction, got one NADH energy carrier is produced. So okay. Now after we form acetyl-CoA, next we can enter Krebs cycle. So this is the overall of the Krebs cycle. So as a biology student, you should memorize all. Memang kena hafal semua punya. Tak ada pilihan. Memang kena hafal. Awak tak ada pilihan. Memang kena hafal. So, okay. So, how to memorize it? We go one by one with the reaction. So, where is the first reaction? So, where is the first reaction? We see where is acetyl-CoA. So, acetyl-CoA is here. So, when acetyl-CoA is enter the mitochondrial matrix, it will react with the oxaloacetate. So this is the first reaction. So the first reaction is known as condensation. So the oxaloacetate and acetyl-CoA will be condensed to form citrate molecule. So if you see here, acetyl-CoA is only have 2 carbon, right? It contain 2 carbon. Acetyl group, right? 2 carbon. And then oxaloacetate, it contain 4 carbon. 4 carbon. 1, 2, 3, 1, 2, 3, 4. So after it condense together, so normally this one, the carboxyl group, this one, this carbonyl group will be condensed with the carbonyl group here. So that it will form this molecule. So the coenzyme A is released. The coenzyme A now is released. And then it will form the citrate molecule. Okay, actually the molecule structure you don't need to know. You just want to know the structure. You just want to know the name only. But for memorization, why it is called as condensation? Because it requires water. They are not water molecule punya. And then it is it will combine together. So normally condensation process normally is combined together, right? Like glucose, not glucose, disaccharide, and then protein, and also lipid. So all of them, if you want to combine the monomer, the process is all called as condensation, right? So same as this, because it is the combined acetyl-CoA combined with the oxaloacetate, so it is known as condensation reaction. And then the enzyme used is citrate synthase. Citrate synthase. Okay. Next reaction is isomerization. Isomerization. So what is it mean? C3 is isomerized to form iso C3. So you see here actually, so meaning that isomerization meaning that the molecular the chemical formula still remain the same, just the molecular structure already changed. The molecular structure already changed. So how it changed? Actually, if you see here, another book is say not say isomerization. Another book say is dehydration and then rehydration. Dehydration and then rehydration. Dehydration is remove. Dia keluarkan satu water molecule, lepas tu dia tambah lagi satu water molecule. You see the Oxford, dia tulis macam tu kan. You see the Oxford, dia tulis macam ni kan. Dia tulis citrate go to isocitrate. Dia keluar, dia tulis it remove water molecule and then add one more water molecule, right? So another process is called as dehydration and then rehydrate again. Okay, but if you want to write this, actually you need to explain like this. If not, you just directly use isomerize itself. Okay, because why it want to remove water and then add another water again? Kenapa dia nak buat macam ni? Because it can form the intermediate. This intermediate cis aconitate. This one it want to form the intermediate because it want to change the molecular structure of this compound. So actually you can see here, I see here, apa ni? You see here, see this structure, it will form the cis structure. Cis isocitrate punya structure. And then after that dia tambah satu water molecule lagi, dia akan form isocitrate again. So the structure you don't need to know, just I want to mention is why it want to remove water and then add water again. Ini sebabnya lah. Ini sebabnya. Kenapa? Kerana dia akan form the intermediate called as cis aconitate. So the enzyme used is aconitase. This is the intermediate. And then after that, you will add water will be added again and then form isocitrate. Okay, so this is the second reaction, isomerization. Next reaction is isocitrate will be undergo oxidative decarboxylation to form alpha ketaglutamate. So if you see this book, see this process, actually it also have the intermediate called oxalate succinate. This one is the book that mentioned in Pelangi. Buku Pelangi, I think, if I am not mistaken. Buku Pelangi, Krebs cycle dia ada reaction ini. Ada reaction ini. So kebanyakan orang cakap Pelangi book buku Pelangi cakap salah. Actually, Pelangi book bukan salah. Just that, but the Pelangi book dia tak ada mention ni. Dia tak ada mention is the intermediate. So that's why Pelangi buku dia macam Krebs cycle dia proses banyak sikit. Compare to the Oxford one, because Oxford just directly give the final product. But if you actually read the biochemistry punya international book, it will give you reaction like this. So in examination, you don't need to know the intermediate actually. You just directly go to the final product. But if you want to write, can. But if you want to write, you need to mention the reaction carefully, the reaction name. So this one actually is oxidation reaction, and then this one is decarboxylation reaction. Two type of reaction. Oxidation, so the isocitrate is oxidized to form oxalosuccinate. This is the intermediate. Just oxidation. So this oxidation will cause the energy carrier NAD plus will be reduced to form NADH. Okay, after that, oxalosuccinate is intermediate, it is unstable, so that it will use the same enzyme again, will be release one carbon dioxide and through the decarboxylation reaction, and then to form alpha ketaglutamate. So this is the whole reaction. But in exam, you don't need to, you can choose it, want to write or not. Either eat If you don't want to write, you just directly isocitrate is undergo oxidative carboxylation to form alpha ketoglutarate. And then got one carbon dioxide is released, and then NAD plus is reduced to form NADH. So just like that, you need to alert lah. Okay, if you want to explain, up to you. Now explain ke tak? Up to you. Okay, and then fourth reaction is about also oxidative decarboxylation also. So this reaction is alpha ketoglutarate will be undergo oxidative decarboxylation and to form succinyl CoA. Dia akan form again succinyl CoA. So this reaction actually same as the reaction in link reaction. Because it already involve coenzyme A already. Dia involve coenzyme A. Dia akan bind dekat coenzyme A. So memang dia sama saja. So if you see here the material that require, you see it require magnesium ion, TPP again, lipoic acid, FAD again. So this reaction actually totally same as the link reaction that you learned like this. Just that the pyruvate changed to alpha ketoglutarate only. But you don't need to expand it. If you want to expand, fine, you can expand it. But for now, you don't need to expand it. Don't need to expand. Tak perlu expand lah. You don't need to expand it. Just that I want to mention you in theory, dia sama saja dengan link reaction punya. So that is why you clearly see here all the components same as the linear link reaction. So the enzyme here is alpha ketoglutarate dehydrogenase complex. Complex. Memang complex kerana dia ada reaction dia sangat complex. Dia ada 3 reaction kan? 3 reaction to form the coenzyme A. But here the coenzyme A is not released lah. Dia takkan release lah. Okay. Okay, so this one is the oxidative decarboxylation also. So the NAD plus also will be reduced to form NADH, and then one carbon dioxide is released. So this reaction same as the link reaction. Fifth reaction is hydrolysis reaction. So the succinyl CoA will be hydrolyzed to form succinate. Dia akan hydrolyze to form succinate. So you see here, the coenzyme is released already. Coenzyme A is released again already. And then one water molecule will be added at here. Okay, so will be added. Okay, so at the same time, our GDP will be phosphorylated to form GDP. But you need to know this one is not energy carrier. GTP is not an energy carrier. Not saying it is not, but it is not a basic energy carrier. Basic energy carrier. The basic energy carrier is ATP. So this GTP will be changed to ATP. So how it changed? So this reaction. So with the help of the GTP, the ADP will be phosphorylated to form ATP. So this enzyme is we call as nucleoside diphosphate kinase. So in exam, Krebs cycle, if you directly say that Krebs cycle produce ATP is wrong. Salah punya. Because the produce is GTP. So this GTP will be further modification again to form ATP. Memang dia akan further modification. So this is the enzyme lah. Nucleoside diphosphate kinase. Okay. Okay, sixth reaction is oxidation. So the succinate will be oxidized to form fumarate. So this one, FAD come out lah. FAD and the FAD as an energy carrier finally come out. So this FAD will be reduced to form FADH2 lah. Okay, so the reaction enzyme involved is succinate dehydrogenase. So you see here, semua senang je kan enzyme name dia. All of the reaction that include NAD, FAD punya memang dia just succinate tambah dehydrogenase. Kalau yang ni dia just tambah alpha ketoglutarate dehydrogenase. Just want to add complex. And then yang ni just Isocitrate dehydrogenase. Senang kan? Okay, so all of the oxidative decarboxylation, oxidation reaction that involve AAD and AD plus, all these just use the substrate name and then add dehydrogenase only, just like that. The enzyme name also very simple. Dia bukan totally not same one. Okay, and then seventh reaction, hydration. So this one it will add one water molecule will be added into the fumarate and then hydration reaction to form malat reaction. So this one enzyme is fumarase, also very easy. Just change the just the T change to S only. Because the enzyme normally is SE one. So just change the substrate name, fumarate, the T change to S only. Okay, and then reaction, malate will be undergoes oxidized to form oxaloate acetate. So form back the origin of the compound lah, origin of the compound. Okay, so this one is oxidation reaction. So this one the energy carrier NAD plus will be reduced to form NADH. So again, also very easy enzyme name, malate dehydrogenase only. So kan? So for Krebs cycle, enzyme name is not very difficult. Normally all of them that involve NAD plus, FAD punya semua just substrate tambah dehydrogenase je. So jangan kata enzyme susah. Tak susah. Dia tak bagi clue awak. So siapa yang cakap susah, ini saya tak tahu lah. You already memorize the substrate and product. So no wonder you cannot memorize this. Just tambah satu dehydrogenase only. So okay. So this is the overall reaction. So after form back oxaloate acetate, it will continue combine with the acetyl CoA and then recycle again. So recycle again. So that is why that is called as cycle, Krebs cycle. It will continue the cycle along the time until you die. Yeah, the cells will undergo this cycle again and again until you die. Kalau orang mati memang tak ada lah. So that's all for the reaction. So want to memorize it. So normally want to memorize it, you can use this stuff. So you need to know where are the NADH produced. So you need to know that the first reaction is after forming the acetyl CoA, we combine with what? The first substrate is what? Who combine with the acetyl CoA? Oxaloate acetate, right? Oxaloate acetate combined with the acetyl CoA, combined with the acetyl CoA through the condensation. So you need to say the condensation reaction to form what? Citrate, right? To form citrate. Form citrate molecule. So this one, the enzyme used is citrate synthase, right? Citrate synthase. After that, citrate will be isomerized to form isocitrate, right? The enzyme used is aconitase. Isocitrate will be undergoes oxidative decarboxylation and oxidative decarboxylation to form alpha ketoglutarate. And then same reaction also, it will form succinyl CoA. And then after that, it will undergo hydrolysis reaction to form GTP, to form succinate. After that, it will undergo oxidation to form fumarate. After that, it will undergo hydration to form fumarase. After that, it will form go through the oxidation to form oxaloate acetate again. So these are all the reactions. So which reaction? Step 3, 4, and 8, 2 NADH molecule NADH are produced. each step. So each step will produce 2 NADH. Why 2 NADH? Because you need to know that one glucose molecule produce 2 pyruvate. So this one I use the one glucose molecule. So 2 glucose molecule. So one glucose molecule will produce 2 pyruvate. So 2 pyruvate will undergo link reaction, so it will form 2 acetyl-CoA. So actually there are two acetyl-CoA, 2 acetyl-CoA equal to 1 glucose molecule. Because so you need to know. So step 3, 4, and 8 is the reaction that NADH are produced. So what is step 3? Step 3 is when isocitrate is undergo oxidative decarboxylation to form alpha ketoglutamate line. Step 4 is alpha ketoglutamate to form undergo same reaction oxidative decarboxylation again to form succinyl-CoA line. And then step 8. What is the step 8? Step 8 is the last step. There are total 8 steps. Last step is oxaloacetate, right? Final product is oxaloacetate. So the previous product is malate, right? The malate will undergo oxidation reaction to form oxaloacetate. So these 3 reactions will involve NAD plus reduced to form NADH. And then was our step 5 is GTP are produced. So if you know GTP, so it will involve succinyl-CoA lah. So the succinyl-CoA is hydrolyzed to form succinate, right? Step 6 is succinate. Succinate will be oxidized to form fumarate, right? Oxidized to form fumarate, right? So FADH2. So they have FADH2. So okay. So these are the main reaction that have produced energy carrier. What? These are the most important one. So overall equation for the Krebs cycle is the pyruvate will be oxidized to release one pyruvate molecule will oxidize to release 3 carbon dioxide. So 3 carbon dioxide meaning there are 3 oxidative decarboxylation at there. 4 NADH means and 1 FADH2 meaning that there are 5 oxidation reaction at Krebs cycle. And 1 GTP meaning that there are 1 hydrolysis reaction at there. So in conclusion, in Krebs cycle, 1 glucose molecule will produce 8 NADH, 2 FADH2, and 2 GTP. This is for Krebs cycle. So Krebs cycle does not produce any ATP. Dia memang tak ada produce ATP. So if you say this 2 ATP memang salah. Salah, terlampau salah. ATP is only produced when the GTP is used to help ADP phosphorylation directly. So alert it. Okay, so that's all about the Krebs cycle. Got one question. What enzyme catalyze each step in Krebs cycle? Yeah, memang kena memorize. Yeah. If you don't want memorize, then question ask no marks dah. Especially structure question. Kalau structure question dia tanya je, tak ada markah, 1 markah. If you think that the 1 mark you want to you want to mendema markah tu, markah tu kepada MPN, up to you. But in syllabus you should memorize it. But I already say the Krebs cycle is the most easiest one compared to calculasis because all the oxidation reaction involve oxidation memang yang ni 1, 2, 3, and 4. This 1, 2, 3, 4 oxidation reaction already very easy one. If you already memorize the substrate, then you should already also also know the enzyme already, directly know, even though don't need memorize, directly know already, because just substrate tambah dehydrogenase. Yang susah maybe yang ni citrate synthase, dan susahnya kok pun senang kan, synthase. Just the difference is synthase and synthase. Kena alert lah, synthase and synthase. Another one yang maybe lah, yang ni susah sikit lah, encodinase. Yang ni memang tak sama kan. But actually for the dehydrogenase punya ada 1, 2, 3, 4 reaction ni awak sudah hafal kan. 1, 2, 3, 4. And also this one, fumarate also very easy. Fumarate hydrated to malate, just T tukar S only. And then what yang ni lah, maybe yang ni lah. Yang ni memang kena hafal sikit lah, kerana memang tak sama dengan substrate dan produk juga. So you can, whatever you want to memorize, there are many ways you can memorize one. So I think all of you will memorize this one, this process. This is the famous one of the famous question. Memang semua orang akan hafal ni punya. In this semester, memang semua awak akan hafal glycolysis, Krebs cycle, and electron transport chain. Memang 100% hafal punya. So I will not really affect this topic punya. So you don't need so normally the people not hafal is chapter 1, chapter 2. Okay, that's all. Okay, so next we go to the ETC, so electron transport chain. So yang ni lebih kompleks sikit. Dia memang complex sikit compared to the glycolysis and Krebs cycle lah. So electron transport chain. So this one is the overall. So we already go to glycolysis, link reaction, and then this one citric acid cycle, also known as Krebs cycle. So why we need electron transport chain? So you need to know that our energy carrier in form of NADH and also FADH2, all of them will go to the mitochondrial intermembrane, go to near the mitochondrial intermembrane surface to undergo to donate the electron, because this one already can be electron, right? NADH, FADH2, and all these two molecules already carry electron. It already is in the reduced form. So it already carry electron. This electron will be very important in electron transport chain. So that is why that is known as electron transport chain. Dia akan involve electron punya. So in the definition of electron transport chain is that it is the series of the oxidation and reduction reaction occur in the intermembrane of mitochondria by transferring the electron through the protein complex. So memang dekat intermembrane punya mitochondria dia ada banyak protein. So there are 4 protein complex, right? Later we will see it. There are total 4 protein complex, and then it will release the electron. So yang ni memang dia akan ada kait dengan chemistry sikit. So for the chemistry, oxidation reaction and reduction reaction, you need to know. Later I will use many terms oxidize and reduce punya. So if you still until now you don't know, totally tak tahu pun apa itu oxidation, apa itu reduction, please refer back your SPM chemistry. Ini SPM chemistry dah belajar punya. Even in your STPM until sem 1, I think sem 1 pun dah belajar oxidation and reduction reaction. Ini memang dia akan kait dengan chemistry punya. So if you still don't know apa itu oxidation, apa itu reduction, memang awak kena go back to SPM. Go back. Okay. This is electron transport chain. So in electron transport chain, because of this electron transport chain punya reaction, it can provide oxidative phosphorylation. So when we go about phosphorylation, actually it will involve the phosphorylation of ADP lah. So this one, oxidative phosphorylation definition is that the main definition is to make ATP from ADP, but how to form the phosphorylation is not directly the phosphate group is not directly come to it, because you need to know phosphorylation of ADP to ATP it require energy. Dia is endothermic reaction, dia kena energy punya 7.38 kilojoule energy. Recap in your chapter 5 first topic is endothermic reaction, dia nak energy. But aerobic respiration kami nak hasilkan energy lah, kami nak hasilkan energy, so nak guna energy again, kami nak form ATP, nak guna energy again, lepas tu form energy again. Tak logik kan, mana logik? Memang tak logik. So how to use the energy, that energy come from kerana memang ADP want to phosphorylate ADP to ATP, memang kena ada energy punya. But it already ATP, takkan nak guna ATP again. Awak nak form ATP, nak guna ATP again to form energy. So memang not sense lah, not make sense. Not make sense. So energy datang dari mana? So energy datang this lah. Energy released at each step of the chain. So in electron transport chain, it will release energy. So what form of energy? Later we will see it. Dia akan release energy. Release energy so that the ADP can use that energy to phosphorylate to form ATP. So this energy is powered by the redox reaction of the electron transport chain. So energy come from where? Like this redox reaction of the electron transport chain. So other electron transport chain, they provide energy in form of energy. So another term is chemiosmosis. So can energy come from where? This term explains this chemiosmosis. Because the energy is stored in a form of hydrogen ion gradient across a membrane is used to drive the cellular work such as synthesis of ATP. So chemiosmosis is the term that explains where energy come from. Want to phosphorylate ATP, mana energy dia? Hydrogen ion. Hydrogen ion gradient. Electrochemical hydrogen ion gradient. So ini akan kait dengan chapter 3 punya core transport. Core transport. Recap again. Apa itu core transport? I already explained it. So in this, I will not explain very detail. Apa itu electrochemical gradient? Itu I assume you already know. Because I already explained it. So for those who absent, maybe you are very smart people, so you don't want to attend my class. Never mind, up to you. So okay. So next, this is the 3 main terms you need to memorize it. Memang awak akan tengok ini 3 perkataan ini. So next we go to the electron transport chain, the protein complex. So if you see here, see this is the structure of the mitochondria that locate in the intermembrane. Intermembrane. So this is the intermembrane space. So this is the inner membrane. You see, if you know that the structure of mitochondria is double membrane bounded, right? So this is the inner membrane. This is the intermembrane space. This one is the mitochondria matrix. So this is the place where the NAD, where all the NAD, the Krebs cycle and zinc reaction occur at here. Memang dia occur at here. So all the NADH and FADH2 that produced from the Krebs cycle will be accumulate at the mitochondrial matrix one. So if you see here, there are total 1 all are the protein complex. So this one is complex 1, 2, 3, and 4. So this is just a general name now. Actually, they are the scientific name also. Later we will see it. What are the scientific name? And then this one, some book, if you refer Biochemistry International book, it say it's complex 5. But in exam, kalau dia keluar, dia awak name it is, you need to say is ATP synthase. So this is one type of the enzyme, that responsible to produce ATP. So, okay, if you see here, apa awak nampak ni in overall, I just in overall reaction, the NADH produced from Krebs cycle and glycolysis and also link reaction, produce these three parts, it will directly go toward the complex one, protein complex one. So it will release the electron to the complex one. Dia akan keluarkan release the electron. So the NADH will be undergo oxidation. It will be oxidized to form back NAD+. And then it will release electron to complex one. Our FADH2 will release the electron, also will undergo oxidation, will release the electron to complex. This complex 1, 2, 3, 4. So for NADH, it will only go through complex 1, ubiquinone Q, and complex 3, and complex 4. For FADH2, it will go to the quinone Q, then go to 3 and 4. So dia tak akan pergi 2 punya. Dia akan pergi complex 2. Dia directly go to quinone Q and then go to the complex 3. Okay. So the electron will be passed through within this protein complex toward the complex 4. Okay, so there are the oxidation and reduction reaction occur within the protein complex punya. So apa itu yang occur, nanti we will tengok. But now I just want to say, okay, when the oxidation and reduction reaction occur within the complex, this will create the electrochemical gradient. The hydrogen ion within the mitochondrial matrix will pump out toward the intermembrane space. Kenapa dia pump out? Kerana dia ada oxidation reduction reaction. So this is one kind of active transport process because the mitochondrial matrix has the low hydrogen ion concentration. So you see here, electron transport and pumping of proton hydrogen ion will create an hydrogen ion gradient across the membrane. So this pumping of the hydrogen ion will create the electrochemical gradient of the hydrogen ion. So all of this, semua ni. Okay, after that, once it already pump the hydrogen ion, it already pump out the hydrogen ion at here, it will directly, the hydrogen ion will be transported down the concentration gradient through the ATP synthase, and then oxidative phosphorylation and chemiosmosis will be occur now down the electrochemical gradient. So this hydrogen ion provide the energy to ADP, so the ADP can undergo oxidative phosphorylation, the phosphorylation process to form ATP. And then the electron for the electron transport chain will be passed through until complex 4. Until at the end, the electron will be accepted by the oxygen, so that it will form water molecule. So oxygen molecule is our final electron acceptor in aerobic respiration. So oxygen molecule is very important in why oxygen molecule is very important to us. Kenapa kami nak memang kena ada oxygen punya? Because of this reaction. Because oxygen act as the final electron acceptor. So if you imagine that if no oxygen, kalau tak ada oxygen lah, memang the electron cannot go to anywhere. The electron will stuck at here. It will directly stuck at here. Kalau dia nak pergi mana? Kerana dia tak ada oksigen, dia hanya pergi kepada oksigen saja. Kalau tak, memang dia akan stuck at here. So if you stuck at here, memang tak ada energy. So memang tak ada energy, memang sel akan mati lah. Tak ada energy, memang sel akan mati. So that is why oxygen is very important in human. So That's all for overall of electron transport chain. This just overall, memang overall je. After that, we will go through one by one, protein complex. Let me say, got one question. Which one? ATP synthase or synthetase involved in chemosis? The enzyme involved is ATP synthase, not synthetase. What are the different these two enzymes? I already forget it, but I think got one book dia ada mention Campbell punya biochemistry dia ada mention it. You can read it, but or later I have time I go to read it again. Mana dia ada mention kenapa apa beza dengan synthase and synthetase. But for now, you just need to know lah. The enzyme that used is ATP synthase, not synthetase. Bukan synthetase. Synthetase dia ada process line. Dia ada process line. So apa process dia, apa beza dia saya tak lupa. But later I check again. Dekat if you want to find it, you can find it in Campbell punya biochemistry. Bukan Campbell yang normal punya, Campbell yang buat biochemistry punya. Ya, ada cakap. Later I found it. Later I explain to you. Okay, next is so now we apa yang berlaku dekat semua ni? 1, 2, 3, 4. Apa yang berlaku? So before we enter here, we will learn many component, coenzyme and also cytochrome. So if you see here, actually within our protein complex, there are also many component. You can see here there are one component called FMN, FES, and also CYT. CYT. So this one is FMN, not FMS. Kenapa saya tulis FNS? FNN. Okay. FNN. So the FNN we known as is flavin mononucleotide. But another the common name is the flavoprotein. So it is one type of the cofactor because it contains flavin coenzyme. It is one type of cofactor. And then FES is that FES, so Fe is ferrous iron, right? S is sulfur. So this one is the iron sulfur protein. So this one is the function is to accept electron from FNN and transfer to Q. And then Q is called enzyme Q or ubiquinone. Which is individually located within the membrane. So if you know that this one Q is individually located one, it can move one. For this protein complex, dia tak bergerak punya. Dia ada bergerak tapi dia tak bergerak macam ubiquinone Q because ubiquinone Q is located individually within the membrane. So dia akan bergerak dari complex 1 lepas tu transfer electron to complex 3. Also it will transfer electron from 2 to 3 also. So and then CYT, what is CYT? You see here there are many CYT, right? CYT is we call as cytochrome protein. It's also one type of protein that contain hand group to accept electron. So these all components play major role in oxidation in redox reaction, oxidation reduction reaction. Okay, now so this is the overall of the process. Memang yang ni just bagi awak macam mana electron dia pergi. So you see here NADH has the lowest affinity for electron, so it will release the electron, it release two electron to FMN, will undergo oxidation to form NAD+. After that, the electron will move, bergerak sampai complex 1, complex 3, semua sini, lepas tu ke sini. Dia akan bergerak pass through at here. For FADH2, it will go to the complex 2 and then go toward the FES. And then you will pass through the electron again until the final electron acceptor lah, oxygen. So dia akan pegang kat sini. So you see here, electron move from an electron carrier with a lower affinity for electron to an electron carrier down the chain with a greater affinity for electron, releasing energy. So you see here, kenapa dia boleh release energy? Because the electron from an electron carrier with the lower affinity of electron. Maksudnya yang ni is lower affinity toward electron. Meaning dia mudah untuk dapat electron. So for NADH, it carry electron, but it has the lowest affinity. Maksudnya electron hanya boleh bind dekat sini. Dia tak boleh bind dekat sini. Dia tak boleh suka-suka bind ke protein complex bind to 444 because it has the lowest affinity. Dia ada lowest affinity. So after that, with a lower affinity electron to an electron carrier down the chain with a greater affinity electron, releasing free energy. So when it go down toward the complex, it will become very greater affinity punya electron. So this one can release the energy. Dia boleh release energy toward this reaction. Okay, now we go through this. So this is the actually more detail about the electron transport chain. Macam mana dia oxidation and reduction. Macam mana dia oxidize again and then reduce again. So you see here all these semua ni, the complex 1, complex 2, complex 3 and complex 4. So macam mana tengok, you see here, I go to NADH first. The NADH as the electron carrier, dia akan oxidize. NADH will be oxidized to form NAD+. So oxidation, it will release electron. Dia release electron kepada FNN, flavoprotein. Okay, it will release the electron to the flavoprotein. So once the FNN release the electron, it will undergo reduction, right? Dia akan undergo reduction to form FNNH2, hydrogen gas. Ada tambah hydrogen lah, kerana dia ada receive electron, right? Electron and also hydrogen. After that, this FMNH2 will be oxidized back to FMN. Dia akan oxidize again back to FMN, but then if got oxidation, it will release electron, right? Dia release electron kepada siapa? FES lah. So dia akan release electron kepada FES. So this is why it say the FESOS. This is oxidized form. So it will, once the oxidized form release electron, it will undergo reduction to form the reduced form for FES. Okay, so dia memang dia recycle punya. So after that, the FES reduced form of FES, it will transfer the electron to coenzyme Q. Dia akan transfer to a coenzyme Q. So coenzyme Q will undergo reduction, reduce to form coenzyme Q, the reduced form of coenzyme Q. So you will see here, dia memang semua macam ni punya. Memang dia akan oxidize, lepas tu coenzyme Q will be undergo oxidize again, undergo oxidation, transfer electron to this FES. And then FES will release transfer electron to CYT-C1, cytochrome C1. After that, cytochrome C1 will transfer electron to cytochrome C, cytochrome A, copper ion, cytochrome A3. So, yang paling penting awak kena ingat is FAM is the first reactant, first substrate that receive electron from NADH. It is the first reactant, first substrate. Ini awak kena tahu lah. After that, it will transfer electron to FES. So, FES, after that, it will transfer to the coenzyme Q. So, coenzyme Q is not the complex. So, actually, complex 1 just only contain FNN and FSS only. So after that, once it transfer to coenzyme Q, so coenzyme Q will transfer to complex 3. Complex 3 also contain FS and also cytochrome C1. After that, it will transfer to cytochrome C again, cytochrome C. So this cytochrome C also same as the coenzyme Q. It is the unit. It is the solid individually locate within the membrane. After that, cytochrome C dia akan transfer electron again to cytochrome A. Cytochrome A. Sorry. Cytochrome A. After that, dia akan release electron to copper ion. After that, it will release to cytochrome A3. So yang paling penting A3 ni awak kena remember. A3 is the last cytochrome that release electron. So after cytochrome A3 accept the electron, it will transfer, release the electron to the oxygen molecule. So the oxygen is the final electron acceptor. So oxygen molecule will be when the oxygen receive the electron and then will be react with the hydrogen ion, two hydrogen ion will form one water molecule. So these are all general in the electron transport chain. So in exam, you should answer based on this. Memang awak kena answer macam ni. Yang ni NADH. So FADH, for FADH punya, dia akan go toward complex 2. So the first component that for FADH2 is not flower protein activity. It is FES. Dia directly pergi FES punya. After that, FES it will transfer electron to coenzyme Q, and then transfer again until last one is cytochrome A3, and then the final is oxygen. Akan accept electron to form water molecule. So this is the overall punya. So in examination kalau dia nak keluar soalan nak explain all this electron transport etc, you need to explain all this lah. So hopefully you guys understand. This just also general one. After that we go to very detail. Memang sangat detail. So apart complex one, so the full name is NADH coenzyme Q oxidobutyrate. Ini full name. Tapi in exam you don't need to write this. If just write complex one is okay already. Ini full name dia lah. Ini full name. So normally the explanation is same as the what I say. Memang based on this. Just ini reaction dia. This is the coenzyme Q punya reaction. You will see here coenzyme Q dia akan undergo oxidation and reduction, right? So when the coenzyme Q undergo reduction process, it will form add on hydrogen atom lah. Two hydrogen atom will be added. Just want to show you lah. Only. Actually, you don't need to know over the structure. All these structure you don't need to know one. Just want to show you only. Okay, explanation same as the what I say before. And then complex 2 is succinate coenzyme Q oxidobutyrate. So this is the full name. So this is the reaction that involved in here. Sama aja, just ini dia keluarkan dekat equation. So sama aja dekat sini. Explanation I also already explained just now. Complex 3 is cytochrome reductase. So this is the full name dia. So normally all these reaction is same as the what we say. Just that got one terms is Q cycle, right? You see here. So what is Q cycle? You know when the coenzyme Q, we know that coenzyme Q will accept 2 electron. Coenzyme Q if go to reduction, dia akan accept 2 hydrogen ion punya. Dia akan terima 2 hydrogen ion. Dia akan terima 2 hydrogen ion. So once it is accept 2 hydrogen ion, so two hydrogen ion. So it only can transfer one electron. In compared to it only can transfer one electron once a time. Satu elektron je, tak boleh transfer 2 elektron punya. Dia hanya boleh transfer 1 elektron. So, when why it only can receive 1 electron because of this cytochrome. Because cytochrome only contain one hand group only. Ferrum ion. Satu je. Dia hanya satu je atom. Satu je. So, maksudnya dekat satu kali, dia hanya boleh accept satu elektron je. Dia tak boleh accept 2 elektron punya. Tak boleh. Dia hanya boleh accept satu elektron. This is a reaction lah. Ferrum ion punya reaction. So, kalau macam ni, coenzyme tu dia ada 2 elektron. Sekarang dia ada 2 elektron. Macam mana dia nak transfer? So if coenzyme Q dia nak release 2 electrons, so one of the electron will directly go to the ferrum FES. How about another electron? So another electron will go to cytochrome B. Another code will be released to cytochrome B, and then back again to coenzyme Q again. So that is what we call as here. So kenapa kami panggil Q cycle here? Kerana coenzyme Q dia nak undergo reaction, dia nak receive 2 electron. Dia memang dia nak 2 electron punya. So macam mana? And then macam mana dia nak release 2 electron at one time? Because in complex tree dia hanya boleh accept 1 electron only. Dan 1 electron nak pergi mana? Bagaimana? So when coenzyme Q, the reduced form of coenzyme Q, dia nak release electron. 1 electron will pergi complex tree. Another electron will go to cytochrome B. Okay. After that, the cytochrome B will release again the electron, go to the coenzyme Q again. Go toward the coenzyme Q again to form this again. Because you need to know that from the coenzyme Q, from the NAD plus punya yang ni pun hanya boleh ada 1 elektron. Dia datang 1 elektron, release pun 1 elektron. Lepas tu nak become stable, actually dia nak 2 elektron. But another electron come from where? Another electron come from cytochrome B. Come from cytochrome B and release to cytochrome B. So that's why we call Q cycle. Ada Q cycle dekat sini. Okay, yang ni saya rasa your school teacher 100% tak ada ajar punya. Memang tak ada ajar. Saya tak percaya your school teacher will teach you this. If your school teacher teach you, memang your school teacher is very expert lah. I can say lah. You need to pray for it or apa. Ada cikgu macam ni. Kalau lah cikgu awak lah ajar yang ni. Kerana yang ni saya pun sendiri buat research punya. Even at that time my school teacher does not teach this. But I hope everyone can understand it. Yang ni Q cycle. Okay. Okay. Another one, finally we go to complex 4 lah. So after we go to complex 3, memang dia akan pergi cytochrome again, same as the oxidation reduction reaction. We will go to cytochrome C. After that, cytochrome C will release the electron to cytochrome A and then go to cytochrome A3 lah. Okay, so the cytochrome A3 will release 2 electron to the oxygen that act as the final electron acceptor. So actually it will only release it will release 2 electron lah. But one by one lah, dia akan keluar sekaligus keluar 1 electron. As I say, cytochrome it only contain cytochrome only consists hem group only, and they can only receive 1 electron. So it will release one by one lah. Kalau dia ada 2 electron, so dia akan pergi the oxygen molecule. Then to form water molecule lah. So the oxygen, repeat again, is the final electron acceptor in aerobic respiration. So in objective question or structural question, dia cakap apa final electron acceptor? Oxygen. Hanya ada oxygen. Okay, so that's all for the complex, for the electron transport chain only. Dan ni kami hanya cakap semua besar ni je. Besar ni je. So, in within the oxidation in this electron transport chain, the redox reaction all at here, it will provide the energy so that the hydrogen ion will be pumped from mitochondrial matrix against the concentration gradient toward intermembrane space. Against punya, that low concentration to high concentration area. Active transport. So you need to know that active transport require energy. So yang ni dia tak guna ATP langsung. Awak tengok dia langsung tak ada guna ATP. So energy come from where? All this redox reaction, all this electron transport chain punya, the redox reaction, that release energy, the redox reaction release energy so that it will directly this energy provide the hydrogen ion pump out to the intermembrane space. Okay, so now you knew that the hydrogen ion will already accumulate in the intermembrane space, already create the electrochemical gradient hydrogen ion at here already. Okay, apart next here. So next we go to chemiosmosis. So we know that the intermembrane space is already have accumulated high concentration of hydrogen ion already. So now this hydrogen ion already create electrochemical gradient, so it can pump down the concentration gradient toward the ATP synthase to produce ATP. So how? Macam mana? Inilah step dia. So you need to know that our ATP synthase, the structure of ATP synthase is one kind of motor. Dia ada rotor. Dia boleh pusing-pusing. Actually, dia bukan dia bukan the protein that biasa awak tengok dekat luar. It look like the motor one. Dia look like it punya. But the structure you don't need to know. You just want to know the process macam mana dia. So once the hydrogen ion will be pumped through this ATP synthase down the concentration gradient to a channel called stator. So dia akan pergi ke sini. Called stator. Okay. Next, once the hydrogen ion go to the stator, it will enter the binding site within the rotor. So the rotor contain the binding site of hydrogen ion. And then it will change the shape of each subunit so that the rotor will spin within the membrane. So it can spin. Okay, once it spins, the hydrogen ion can make one complete turn before leaving the rotor and passing through a second channel in the stator into the mitochondrial matrix. So once the hydrogen ion is complete the turn, and then it will leave the rotor. Passing through the second channel of the stator. So this is the second channel of the stator. Then it will go toward the mitochondrial matrix lah. So this is the flow of the hydrogen ion. For hydrogen ion akan pergi stator dulu, then go toward the rotor, and then after spin one time, after that hydrogen ion will pass through the second channel of the stator, then back to the mitochondrial matrix lah. Okay, this spinning is very important. Why? Because this spinning will cause an internal rod yang ni macam pipe punya, tube punya rod to spin as well. Dia ini spin, memang dia pun akan spin lah. Dia macam dah kait-kait relation punya. Dia akan spin juga. Lepas tu, this rod will extend like a stalk into the knot below it, which is held stationary part of the stator. So like that. So it will form the knot like that. So dia akan bengkak lah. Yang dekat bawah punya dia akan bengkak. Yang actually dia akan kus tak akan bengkak macam ni punya. Dia akan macam ni punya. Akan look like this one. Memang kus punya. Lepas tu dia akan bengkak. So it will form the knot lah. So the bengkak punya terms called knot. So this one turning of the rod activate the catalytic site in the node that produce ATP. So yang ni, this is very important because when the node is already spin, the node is bengkak, called as form the knob, so that the ADP can be undergo phosphorylation to form ATP. So inilah overall process dia. Memang kena hafal lah. Saya tak boleh buat apa. Just that you need to know lah all the process. But the in general process is that hydrogen ion will pass through the ATP synthase, and hydrogen ion will provide energy to the ATP. So it can form ATP. Okay, so I think that's all for today. Memang cerita panjang kan? Yang ni memang dia semua very difficult. Understand. I don't know you guys understand or not until now. So in general, I just repeat again. In general, just that electron transport chain, you will see clearly. Imagine it, just electron punya. Dia transport electron to complex 1 until pergi ke coenzyme Q, lepas tu pergi 3, lepas tu pergi 4. Lepas tu elektron pergi mana? Finally dia pergi oksigen. Okay, kenapa dia nak buat macam ni? Kenapa elektron nak transport macam ni? Because got oxidation reduction reaction. So this reaction will provide energy. Kenapa nak energy tu? Energy tu to provide proton pump, hydrogen ion pump. To pump the hydrogen ion from mitochondrial matrix to intermembrane space. Kenapa dia nak pump out? Because it want to create the electrochemical gradient. Kenapa nak create electrochemical gradient? Because it need to produce ATP. Siapa yang produce ATP? ATP synthase. So, ATP synthase macam mana provide ATP? Inilah hydrogen ion. The hydrogen ion will move down the concentration gradient toward the ATP synthase, then it will provide energy to ADP. So ADP can undergo phosphorylation to ATP. So actually, the final result, the final objective in electron in ETC is to produce ATP only. Senang je. Kami nak ATP je. Tapi proses dia sangat kompleks. Kami dah produce ATP, proses dia semua ni memang sangat kompleks. So that's why all that did. So this topic that's why it is very difficult, but I think that all of you will take more time effort for this kerana dia susah kan. So semua orang akan take time at here. So whatever, so any question? So for now, for today, I teach until here lah. Saya tak mahu cakap banyak-banyak lah. I teach until here. Nanti awak semua sakit kepala kalau awak saya continue. Next, tomorrow I will explain one glucose molecule produce how much of ATP, and also the other topic of aerobic respiration lah. The carbon monoxide effect, cyanide effect, and also the alternative resources, how protein and lipid provide the alternative resources to produce energy. All that lah. So that's all for today. Okay, so if you have any question, you can ask it. So now I try to find the difference. You say that what are the difference in synthase and synthetase? Let me check it. Kalau saya tahu lah, I will try to check it. Yang ni. So you guys will any question, you just ask it. For a moment, I try to check the. Hmm. Honey, okay, so I already find the answer. Uh, the difference between synthase and synthetase is that synthase enzyme does not require energy for phosphate bond hydrolysis, and synthetase need energy. So the meaning is that the energy come from where? So normally synthase is the enzyme that do not that the enzyme, it does not require energy from phosphate bond hydrolysis one, but synthetase require. So that's why we refer to the Krebs cycle punya. Awak ada nampak kan? Krebs cycle punya dia ada dua enzyme berbeza dan synthase and also synthetase. So synthase yang ni dia tak perlu energy punya. Dia langsung tak perlu di energy. But synthetase it require energy from the phosphate group. So ini beza dia lah. So beza dia lah. Okay, so hopefully answer your question. Type in the chat box. So I think no question because kerana banyak lah. Okay, so tak mahu cakap banyak-banyak. So that's all for today. Thank you. Bye.